You know, for pretty much all of human history, there was this vast, invisible universe existing right under our noses, totally unseen. A world that, it turns out, holds the secrets to life, to disease, to, well, everything. This is the story of how we finally built the eyes to see it.
I mean, what if you could actually peer into a hidden dimension that's teeming with life, where just a single drop of water becomes this bustling, chaotic metropolis? This wasn't some sci-fi fantasy. It was the real question that kicked off a scientific revolution that's been going on for 400 years.
Just imagine being the first person to actually see it, to put a drop of pond water under a lens and discover not just water, but thousands of bizarre swimming creatures. That's what the very first pioneers of the microscope saw. They called them animalcules, little animals, because what other word was there for what they were witnessing?
And this new way of seeing revealed something even more profound. I mean, it didn't matter what they looked at, a leaf, a fish, a person. Under enough magnification, they all shared these fundamental repeating structures. This was the very beginning of our journey to understand what we're all truly made of.
So who were these people who opened that very first gate to this invisible world? Well, it all started, maybe surprisingly, not in some fancy university lab, but in a humble spectacle maker's shop.
So back in the 1590s, a father-son duo, Hans and Zacharias Janssen, were just messing around. They put a few lenses together in a tube and, voila, things looked bigger. Their first compound microscope only had a magnification of about 9 times. It was really more of a toy, a fun novelty, than a serious scientific tool. But hey, it was a start.
Okay, fast forward a few decades. An English scientist, Robert Hooke, takes this basic idea and just runs with it. He publishes this incredible book, Micrographia, that shows the world what a better microscope could really do. And when he looked at a super thin slice of cork, he saw this structure that reminded him of the small rooms in a monastery, which were called cella. So he called them cells, and, well, the name stuck.
But the true rock star of this whole era, he wasn't a professional scientist at all. His name was Antonie van Leeuwenhoek, and he was a Dutch draper. His day job was selling cloth, and his hobby was grinding these tiny little lenses to check the quality of thread. But his insane curiosity and incredible skill were about to change science forever.
Get this. While Hooke's fancy compound microscope could manage maybe 50 times magnification, Leeuwenhoek's tiny, simple, single-lens devices, we're talking basically a ridiculously powerful magnifying glass, could get up to an astonishing 270 times magnification. He had unlocked a level of detail that literally no one on Earth had ever seen before.
And this just shows you how fast things started moving. Within just a few decades, we went from that simple little toy to Leeuwenhoek discovering actual bacteria and seeing our own red blood cells. This complete explosion of new observations is what led directly to scientists like Schleiden and Schwann proposing cell theory in the 1830s, that fundamental idea that all living things are made of cells. But just as this incredible new world was opening up, scientists slammed right into a wall. A hard physical limit that basically said, you can see this much, but you will never see anything smaller. So this wall was called the Abbe diffraction limit. It was figured out by a guy named Ernst Abbe way back in 1873. And here's the basic idea. Because light itself travels in waves, you just can't use it to see details that are smaller than about half of its wavelength. Think of it like trying to feel the tiny grooves on a vinyl record while you're wearing super thick winter mittens. Your tool is just too big and clumsy for the fine details. Now for the light that we can see, that hard limit is about 0.2 micrometers, or 200 nanometers. And for almost a century, that was it, the end of the road. Anything smaller, a virus, a single protein, the actual machinery inside our cells, was just doomed to stay invisible forever. The first gate was open, but the next one seemed permanently locked.
But, we humans are pretty clever. If light wasn't sharp enough, we figured, what if we could find a whole new kind of light? Something with a much, much smaller wavelength.
And this was the absolute genius of Ernst Ruska and Max Knoll. In 1931, they had this radical idea. They realized that a beam of electrons could be focused using magnets, in pretty much the same way light is focused with glass lenses. And here's the kicker. Electrons have a wavelength thousands of times shorter than light. So in theory, they could see things that were thousands of times smaller. Ruska would eventually get a Nobel Prize for that incredible insight. And just look at the difference here. We went from a max resolution of 200 nanometers with light all the way down to 0.2 nanometers with electrons. That's not just a little better, that is a 1,000-fold improvement.
A second gate to the microcosm had just been blasted wide open. We could finally see viruses. We could see the super intricate machinery inside our own cells. But of course, with all this incredible new power came a new wall. The intense electron beams and the vacuum needed for these microscopes would instantly kill and destroy any living sample. So we could see the detailed blueprints of life, yeah, but we couldn't actually watch the machinery run. So how could we possibly bridge that gap?
The solution, which really came into its own in the early 2000s, was just pure genius. It didn't break the laws of physics, it just found a brilliantly clever way to sneak right around them.
And here's the core idea. That Abbe limit, that physical wall, it really only applies when everything is lit up at the exact same time so it all blurs together. But what if you could just turn on the lights for one tiny little molecule at a time, figure out its exact location, and then turn it off before you light up the next one?
So here's how it works. Scientists attach these special little fluorescent tags to the specific molecules they want to see inside a living cell. Then, using lasers, they flick on just a handful of these tags, making sure they're all spread really far apart. Now, each one still glows as a blurry spot, but because it's all alone, it's really easy to mathematically pinpoint its exact center. Then, you just do that thousands and thousands of times over, each time lighting up a different random set of molecules. Then you layer all those precise center points together to build one final unbelievably sharp image.
This incredible technique, known as Super-Resolution Fluorescence Microscopy, finally let us waltz right past the Abbe limit. For the first time, we could watch the actual dance of molecules inside living, breathing cells. And for this absolutely stunning achievement, its pioneers were awarded the 2014 Nobel Prize in Chemistry.
You know, Ernst Röske, the father of the electron microscope, really said it best. Each new type of vision, each new way of seeing, it opened another gate, revealing another layer of reality that had always been hidden from us.
So from Leeuwenhoek's little animalcules all the way to watching the real-time choreography of proteins, this whole 400-year journey has been about opening one gate after another. These new super-resolution techniques, they're our third gate, and it really makes you wonder, what secrets are just waiting for us behind the fourth?
Hey, if you enjoyed opening these gates to the unseen world with us, help us unlock more by hitting that like button. And make sure you subscribe for more explainers that explore the hidden ideas shaping our universe. Thanks for watching.